all four of us were supposed to be talking about some component of DNA modification, but it'll just be the three of us today, so we're going to miss out on a little bit of knowledge about cloning. But we are discussing changes into human beings in an irreversible and potentially life-changing way. The idea that the genome could be modified has been in the scientific community for a while, both in the popular science for probably even longer than it was possible in the professional sense. But it wasn't until just 4 years ago that we found a truly, what I think is a paradigm-altering method to change the human genome almost completely at will. It used to take a PhD thesis, an entire PhD thesis, to figure out how to change a single gene in maybe like even a lab rat. But now we can do so almost pretty cheaply, about $2,000 for certain genes, not in humans yet. But unsurprisingly, the patents for this technology, though, are extraordinarily valuable and may even be priceless. And the power to change the course of human evolution with unerring precision requires the discussion that involves all of us, because that's something that impacts all of us, that it impacts the future of what it means to be a normal human being. If everybody has access to genetic modification, it may mean that every single human being could be 7 feet tall and live 200 years at least. And that's something that impacts all of us and that we should all have a voice in. So the purpose of my section of the presentation is to make you think about the kinds of questions that you need to have answered for yourself before you can enter in a debate about if there's ever a bill before Congress about how should we be restricting the use of human genetic modification. If there's ever a discussion to be had, these are the kind of things that you need to think about. And I think that we do that effectively if we give ourselves a foundation to stand on that will enable informed discussion about it, about the implications and the consequences that come along with fiddling with the human genome. So CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. And that doesn't mean a whole lot to me, or I'm sure I could have figured it out, but that just means that in prokaryotes there are codes in their DNA that combat the DNA of something like a virus that invades it. So some viruses infect bacteria and stick part of their own DNA into the DNA of the host. This is how HIV works in human beings. This is why you get it and then symptoms disappear for a long time. They infect your cells and hide themselves in your own DNA and then come back to life, so to speak, later on. So bacteria obviously didn't really enjoy having their genome being taken over. This isn't good for them if eventually the virus busts out of the cell and kills it. So what they did was they mutated what is called the CRISPR gene that encodes for a protein called Cas9. And Cas9 is able to cut bits of DNA out that are harmful to the cell. So if HIV sticks in something that's bad for the cell, it can find if it doesn't belong there, it can find it and it can cut it out and just stitch things back together. And that's how it avoids death by virus. And that's how human beings can use it too. And the cool thing is that it encodes for a protein, the Cas9 protein, and if we just pair any sequence with it, it'll cut that out of anything that we inject it into. So say that there's a gene that'll make you only 5 feet tall if you have it. And that's the tallest you can grow, and it's just a single gene. If we know what that sequence is, we give it to Cas9 and inject it into the person, and it will cut out with 100% precision only that gene and leave you, and it can be replaced, it can be taken out completely, or it could be altered in some way. So the existence of CRISPR was established in the 80s by Yoshitsumi Ishino, but he didn't really know what to do with it. And it wasn't until just 2012 that Jennifer Doudna at UC Berkeley was able to modify the genome of a prokaryote, something without a nucleus. And then Feng Zhang and his team at Harvard did it in a eukaryote not long after that. So they both have patents on the use of CRISPR. But CRISPR isn't the only gene modifying technique, so when you consider, should we even be modifying genes, is that a human thing to do? We've been doing it for millennia. And you'll hear about other methods or ethical questions from my other group members, but some examples of how we've already been doing it are dog breeding, selecting corn plants that'll make corn grow bigger and taller, choosing strawberry plants that make strawberries more red. We've been modifying the genes of things for so long, and it's all just by choosing what kinds of things get to reproduce. So this is not the first time that we've been modifying the genome, but it's definitely the most advanced and specific, and it really could change the course of human life. The first time humankind purposefully manipulated genes was in 1974, and then 20 years later, we got the tomatoes that you see on the left. Those are the flavor saver tomatoes in the group under gene silenced. Tomatoes have a gene that encodes for an enzyme that makes them spoil. So after a while, tomatoes produce an enzyme and it makes them spoil and they will degrade. But for the flavor saver tomatoes, which were a big controversy in the 90s, they took that gene out. Not with CRISPR because it wasn't around yet, but using another technique. They just removed it completely. So now it doesn't make this enzyme, and they lasted, as you can see, far longer. They're still not immune to oxidation or to environmental effects, but even after 45 days, the tomato looks decently edible. And today, we're at the point where you can buy, I think they did this using CRISPR, you can buy a fluorescent fish for $10. So this fish was not originally neon green. But now we have the capability to take out, switch, or add any genes that we want to. We have to consider what the catches are. And there are quite a few of them. The biggest one in my mind is that we don't always know what the consequences of switching a gene is. If you want to go ahead and switch the slide, this is neurofibromatosis type 1. This is obviously a skin disorder in which you get extraordinarily painful growths from the nerves of your nervous system. It is encoded by a single gene on your 17th chromosome, and if we know that you are at risk before you are conceived, with CRISPR, presumably, we can delete it right out of your genome, but it has to be before you are conceived, otherwise all of your cells are going to have it and we can't possibly change the genes of all your cells. So any modification we make with CRISPR is pretty much dependent on you not being even conceived yet. We have to consider the consequences of that though. What are the consequences of that gene deletion? In the case of neurofibromatosis, it's unlikely that there are any negative consequences especially that could outweigh the positive benefits of not being in pain constantly throughout your life. But what about for genes that are somehow less necessary, or gene changes that are less necessary than something that is quite painful? You'll hear later about designer babies, or modifying the genetics of your future child to give them whatever trait you want. But imagining a situation somewhat less necessary than neurofibromatosis, but a little more necessary than giving your child genes that will turn her into a professional weightlifter. So if we can imagine that scientists find a gene that's linked to obesity, but not the gene that causes obesity because such thing doesn't exist, but one that all obese people have or very many obese people have, and it's not found in a whole lot of average people. Should we remove that gene? Questions to ask as both a philosopher and as a scientist are, does the possibility of a benefit, which could be the reduction in obesity, outweigh the risks, which are that we don't necessarily fully understand what happens when we remove a gene? A lot of traits that we have are encoded for by multiple genes or by the interactions of multiple genes. So taking one out that causes obesity, that we think is linked to obesity, that gene in those obese people for some reason may have been linked to the development of your eye. So removing the obesity gene could prevent you from being obese, but your eyes don't develop properly. What if it was necessary to develop even the brain? Is obesity so big a problem that we are willing to risk the fact that we don't know what happens when this gene comes out? We don't know that its interactions with other genes are necessary somehow for human life. Even if we take it to a personal Kantian level, if we're not going for benefits outweigh the risks, it's really not that simple. Because for one thing, the person can't choose whether or not that they want to be modified because we have to do it before conception. So that brings the question, do they have rights to their body before they are even conceived, before they are even born? And then nobody understands all the potential risks. So even as an individual, you can't understand the consequences of the decisions you're making, but in the first place, you are not living, so you can't make that decision anyway. So if we are modifying the genome of future children before they're even an embryo, we are making decisions that aren't their choice. Are we making decisions that should be best left to them? Consider a scenario that you and your spouse have talked to a genetics counselor, and you have a 1 in 4 chance of having a kid with cystic fibrosis, which is a single gene disorder. if it causes a buildup of of mucus in the chest and the lungs, and it's so bad that you require percussion or something to hit you in the back to knock it loose, and your life outcome is maybe 30 years at best. Although there are other treatments available as we progress medically, but would it be right? Would you feel comfortable from, especially a Kantian standpoint, from essentially removing that gene from the child without their consent? Because we want to say, for a child, a lot of the times, parents do have the authority to say, I want this done to my child, but this is before they're even alive. And the average expectancy being 30 years, would you feel comfortable saying, which I feel like Kant might have said, is that the kid should be allowed to exercise autonomy over themselves. So we don't take the chance to remove that deadly gene from them because it wouldn't be their choice. And that's really where it gets kind of hairy. And then how about for height? Would you feel differently about height, the height gene, than you would about cystic fibrosis? If we could identify a gene that would make your kid a shoo-in for being on the basketball team, would you feel comfortable giving that a try? The same questions come into play as they did earlier. Do they have autonomy over what happens to their own bodies? Before they're born, do the benefits outweigh the risks? The possible benefits outweigh the potential risks that we may or may not be able to identify. And I think we'll find a lot more people are uncomfortable with the idea of making somebody taller than they are with editing a gene that could kill them. And I think we'll find, because you're now giving a kid, let's say in the case of height, an advantage over other people. If that person had access to genetic modification, they can be 7 feet tall because of a gene modification. They've got what some may consider an unfair advantage. But I would like to think of a little bit about Kehlani's ethical dilemma with performance-enhancing drugs, but with a new twist, because now she was considering whether it was fair or right for people to take performance-enhancing drugs in a lot of different ways, but one of them was it kind of takes away from the spirit of the game. Could they really have done that without use of the drugs? But I found that, or in thinking about it, there have been people in sports that have always had an advantage over people than those who were not like them. For example, my older brother is 5'6", and no matter how hard he worked, he would never be in the NBA. And Yao Ming did not exercise any bodily autonomy to choose to become 7'6". His hustle didn't give him the ability to reach up to the rim and touch it without jumping. And was that fair to those whose height kept them out of professional play? The technology has the ability to level the playing field. If we can find a gene that makes someone 7'6" like Yao Ming, wouldn't it be more fair in that sense for the people who didn't choose to become that good because they're tall? The people who aren't modified, however, might be left out in the dust. And that's why we have to consider the social impacts of it. For example, approximately 50 to 80% of your genes probably determine your intelligence. So although an environmental factor can absolutely blow that out of the water, in some cases, most of the time, you are as smart as you are, and that is determined by birth because of your genetics. So what do we think about becoming genetically modified Einsteins? What happens to the people who aren't genetically modified Einsteins because we can't change our intelligence genes later on in our lives? It has to be done pre-conception. So if access to this sort of technology becomes expensive, then it will create the greatest inequalities that the world has ever seen, because only the rich and famous will be 7'6" Einsteins playing for the NBA and making even more money, because not everybody will have access to it. If the question becomes, how should CRISPR be dealt with? I think that the answer is that it should involve all of us, no matter what. Is that we all have a stake and we all have voices to draw the lines where they think they should be drawn. And I think that came out kind of intuitively. Is it okay to modify somebody to be 7 feet tall versus can we take out the cystic fibrosis gene from this person? If everybody was engineered to be more happy more often and to suffer less and to have greater pleasures and experiences in life, I do think that's worthwhile though. Even though that's not necessary to live, that is what we all consider to be a life well lived. If we can give you a gene that gives you more dopamine when you have good experiences, you'll just lead a better life. And I think that's worthwhile. Getting it right to the point where we can give people more pleasure and to have better experiences, but also avoiding the potential scientific drawbacks of we don't know what all these gene interactions are and the inequalities it could produce. It's a very complex, and it's something that's going to take all of us, and I think we should all be informed about. Thank you.